Hi and Assalamualaikum warahmatullahi wabarakatuh. My name is Siti Nomaisarah binti Rostan. As first presenter, let we discuss for our group research. Let we start with introduction. The virus CHIKV is a mosquito-borne virus belonging to the alpha virus genus in the Togaviridae family. It was first isolated in Tanzania in 1952 and is endemic to Africa, Indian and Southeast Asia. In Africa, CHIKV is maintained in a site-weighted cycle. Involves white mosquito that preferentially feed on premix. In Asia, the virus is primarily transmit in a urban cycle through human biting. Mosquito particular Aedes aegypti and Aedes albopictus. In particular, CHIKV 0.515 replication was restricted compared to CHIKV 0.6.1 in these mosquitoes. Both CHIKV 0.511 and 0.6.2 compare similar pattern for invasion into the salivary gland, suggesting that this tag was not the key factor in determining susceptibility. Overall, the finding of the study provide insight into the differently susceptibility of various mosquito population to CHIKV isolate. Understanding the variation in susceptibility among mosquito species and population is crucial from predicting and controlling CHIKV outbreaks.
Okay, now for the mosquitoes part, the collections and hearing of AE albopictus mosquitoes from LA Reunion and Myotic in 2006, and these mosquitoes in chikungunya virus infection was investigated and compared to other mosquito strains. Okay, for the viruses, these points describes the isolation, characterization and experimental procedures related to chikungunya virus strains used in the studies. Oral infection of mosquitoes. These points outline the experimental procedures carried out to assess the viral titer, discrimination rates, vector competence and viral replication in the AE albopictus mosquitoes used in the studies. Histological examination is to provide valuable information about the structural and cellular changes in mosquitoes infected with chikungunya virus, helping researchers understand the viral's impact on the mosquito vector. Quantitative RT-PCR analysis is to measure the number of chikungunya virus RNA copies in infected mosquitoes. The process involved RNA extraction, generation of standard curve, RT-PCR amplification, melting curve analysis, and data normalization to quantify the RNA copies.
The result of study provide the study review the susceptibility and vector competence of Aedes albopictus mosquito in LA Reunion and Mayotte Island to CHIKV virus infection. The STD and F2 mosquito population show increased susceptibility to CHIKV strain code 0.6.21, while strain 0.5.115 had a lower viral titer. Dissemination rates varied among mosquito population and strain with strain 0.6.21 showing higher rates. Vector competence show different in replication efficiency between strains with some female replicating the virus as efficiently as other. Histological examination confirmed viral colonization in the midgut, salivary glands, ovary, and central nervous system.
Manometry 22115150. Today I'm going to talk about discussion for this thesis. 2005 until 2006, massive chikungunya outbreak in La Reunion Island affected 1/3 of the population. Chikungunya previously circulated in Africa and Asia but not reported in La Reunion. Study vector competence of chikungunya in different mosquito including albopictus from La Reunion. Finding chikungunya chikungunya 06.21 strain had higher dissemination rates and replication in albuterol from Laurinen. Miket question for chikungunya replication. Albuterol from Laurinen as efficient vector as Egypti and albuterol from Vietnam when infected with chikungunya 06.21. Chikungunya 06.21 strain spread more effectively in albuterol mosquitoes from Laurinen compared to chikungunya 05.115. Infection and replication of chikungunya occur in the mosquito miket as salivary glands. Chikungunya 06.21 replicates more effectively and uniformly in the miket compared to chikungunya 05.115. Once chikungunya crosses the miket barrier, both strains replicate similarly. Chikungunya can be transmitted by albuterol as early as 2 days after feeding on affected blood meal. The short incubation period of chikungunya may contribute to its rapid spread in Laurinen Island. Chikungunya enter a region with a low immunity and lots of albuterol mosquito. The outbreak involved the same chikungunya strain in the multiple island. Egypti mosquito transmitted chikungunya better than albuterol. Chikungunya 05.115 may be the original form of the virus in Laurinen and had mixed result in albuterol in Laurinen, mainly feed on human, increasing the risk of transmission. The E1226B mutation is chikungunya 06.21 likely helped its spread to albuterol. Tourism contributed to rapid spread of chikungunya. Further research is needed to confirm the role of the E1-A226B mutation in chikungunya emergence.